The assessment of arthropod abundance in crops is critical for investigating population dynamics of species and species interactions. However, acquiring reliable population estimates that reflect the true abundances of arthropods in the field is challenging. Here we describe the modification and application of a leaf blower-vac for suction sampling of arthropods in rice. In the first section of this video, we will explain the stepwise modification of a leaf blower-vac for suction sampling. In the second part, we will explain how to use the blower-vac sampler in the field, and in the third part, we will show the sampling results. First, we need to collect all parts that are necessary for the modification. For this video, we used an Oleomag BV300 leaf blower-vac that is commercially available. PVC pipe 1 has a diameter of 110 mm and will be connected to the blower-vac. PVC pipe 2 is a standard 110 mm diameter pipe and will be used to connect PVC pipe 1 to PVC pipe 3, which will reduce the pipe diameter from 110 mm to 50 mm. PVC pipe 4 is a standard 50 mm diameter pipe and will be used to connect PVC pipes 3 and 5. PVC pipe 5 will further reduce the pipe diameter to 32 mm. PVC pipe 6 will enlarge the diameter of the mouth part from 32 mm to 63 mm. PVC pipe 7 is a standard 63 mm diameter pipe, which will form the suction mouth of the machine. The end of this pipe will be connected to the hose. The hose should fit the diameter of pipe 5 and 6 and should be about 1 meter long. It should be reinforced with metal because it has to withstand the